Hi Vignesh, we can wait for 2-3 minutes more and then we can start.
Okay, ma'am.
Hello everyone. So I think we should start it now. So welcome to the last discussion session for this functional genomics course. And today I'll be discussing taking the doubts on the week 4 session in which Professor Ganesh basically discussed about the comparative genomics. So can someone please confirm are my slides visible? Okay, thank you. So, firstly, I would like to start. I think many of you have the doubts in the technique, chromatin immunoprecipitation technique. So, firstly, I would like to take up this technique. So, what does chromatin immunoprecipitation technique means? As the name suggests, it basically this technique focuses on the chromatin and utilizes the immuno, that is the antibodies, for the precipitation of this chromatin molecule, chromatin, which is condensed or in uncondensed form, depending on what form it is present. And then we analyze the DNA which we have precipitated, or I can say which we have immunoprecipitated using antibodies. So the figure here demonstrates that it is a cross-linked chromatin, and firstly we isolate and shear the chromatin mechanically, and then we add the antibodies which are specific for acetylated and terminal histone tail. So the use of antibody basically depends on what is your purpose. So in this figure, the purpose is to check the dynamics of the chromatin, whether it is acetylated or methylated. So in this, we have used the antibody which are specific for the acetylated and terminal histone tail, and therefore it will only bind to the residues which are acetylated. So when we incubate this chromatin, which we have isolated and which we have fragmented with the antibodies against the acetylated histone and terminal tail, then we can further immunoprecipitate this complex, and by using the enzymes like proteinase K or various other reversible cross-linkers which we can use, we can release the DNA which we have immunoprecipitated, and we can analyze this DNA using the techniques like PCR. So, not only this, we can use the antibodies against the different proteins where we want to see whether these proteins are interacting with the DNA or not. and what are the regions with which these proteins are interacting in the DNA. For example, if you want to see the transcription factor or if you want to see the binding sites for the enhancers or the repressors, we can have the antibodies specific for those, and then we can immunoprecipitate our DNA, and because the antibodies will bind specifically to the site where our enhancers or the promoters are present, and then we can analyze the segment of the DNA to which these enhancers or the promoters bind. So in this way, chromatin immunoprecipitation techniques basically helps in analyzing the state of the chromatin state, and also helps in identifying various sites in the genome. So this is a very easy question, representing the figure somewhat similar to in the previous slide. So shown below is a schematic of an assay which is used for genomic approaches, and which one of the option correctly identifies the method that the schematic represents and its objective. So whether it is immunohistochemistry that helps in the identifying the localization of the proteins, or it is fluorescent in situ hybridization that helps in the identifying the localization of genes on the chromosome, is it chromatin immunoprecipitation technique that helps in understanding the chromatin dynamics, or is it Western blot that helps in the nuclear in studying the nuclear fractionation? So this is very easy. If any one of you, yeah. So most of you have answered it. So it is option C as you correctly answered. The next question is, chromatin immunoprecipitation method can be used to identify the regions of the genomes that are transcriptionally active, that is the chromatin dynamics, whether it is acetylated or methylated. Second is region of the genome on which to a given transcription factor may bind. Option C, region of the genome onto which repressor binds to, and option D is all of these. So this is also very easy, right? So Divya has answered option D. Anyone else who would like to answer or who disagree that it should not be option D or something else? Okay. So correctly answered. The answer is option number D, which is all of these. So as I mentioned earlier also, using chip assay, we can identify the regions of the genomes that are transcriptionally active. We can identify the site for the binding of transcription factors or the site for the binding of enhancers and the promoters. So the next concept is comparative genomics. Comparative genomics is basically a field of biological research in which researchers uses a variety of tools to compare the complete genome sequences of different species. And by carefully comparing the sequences from the different species, they can pinpoint the regions of similarities and differences, and based on this, they can have an idea about various features which are present in an organism. Suppose we are, it can tells us about whether we are evolutionary, whether a particular gene or a protein is evolutionarily conserved, and if it is evolutionarily conserved, it is supposed to have a very important function. That is why after the natural selection, after under the selection pressure, it is still conserved in most of the genomes. So what is comparative genomics? So researchers choose this comparative genomics to study or to compare the different genomes or of various organisms, and basically it helps in answering the time scale of evolutionary conservation. What are it also tells us what are the common features of different organisms, such as if we are talking about humans and fish, so what are often encoded within the DNA evolutionary conserved between them. It also helps us in looking at closely related species such as humans and chimpanzees, and shows which genomic elements are unique to each. So it also helps in finding the differences basically. And genetic differences within one species such as our own can reveal variants, and therefore these variants may have an important role in context of the diseases. And therefore by comparing the genome of one individual which is supposed to be a healthy individual to the genome of an individual who is suffering from some disease, we can identify whether there is a role of any genes which is responsible for causing this disease or not. So in order to understand the differences, or in order to compare the genomes of various organisms, we need some bioinformatics tools. So bioinformatics research basically merges the field of biology with a mathematical or computational programming. Basically, the most important concept in the field of bioinformatics is the sequence alignment that helps in comparing the genomes. So, sequence alignment is the main process in bioinformatics, which is used for measuring the similarity or the differences between the biological sequences. It may be a DNA sequence or it may be a protein sequence. Since all of the organisms in the having the DNA, since the DNA in all the organisms is composed of ATGC, but the composition or the alignment or the sequence or the series in which this ATGC is present is different in from one species to another species, thereby leading to the production of different proteins. So this can be found by aligning the sequences from the multiple species. So in case if this sequence alignment can be divided into various types, like it can be a multiple sequence alignment. As the name suggests, it is generally the alignment of three or more biological sequences of similar length. And from the output, homology can be inferred and the evolutionary relationship can be studied. By contrast, or on the other hand, pairwise sequence alignment, as the name suggests, that it involves a pair of sequence. So these are the tools that are used to identify the regions of similarity that may indicate the functional, structural, or evolutionary relationship between two biological sequences. In order to align the sequences, we can either align them in a global alignment fashion where we uses the alignment tool in order to align the complete sequence from starting to the end, and we try to find the different regions where the alignment can be found. While in case of local alignment, we just this local alignment tools tries to find one or more alignments describing the most similar regions which are present between the sequences to be aligned. So this is then schematic of a global alignment versus a local alignment. So the upper one is the global alignment. Global alignment finds the alignment over the entire length, as I mentioned in the previous slide, by aligning the matching regions of the two sequences. And here in this diagram, we can see that in this figure, we can see that the matches are shown by the blue color stretches. And this fragmented region here is basically the gaps which are introduced in the sequences in order to align or in order to find the best optimal alignment. So while in case of local alignment, the local alignment tries to find the common subsequences between two sequences with high alignment score regardless of what is the length of the other sequence. So is it clear what is global alignment and what is local alignment? Okay. So next is the tools which are used for the sequence alignment. So for multiple sequence alignment, we can use various tools which are available. Like for example, we can use Cluster Omega. We can use tools like Cons. We can use K-Align. We can use MAFFT, or we can use tools like Muscle. Similarly, for global alignment, we can use which is based on the Needle-Wunschmann algorithm. So we can use tools like Needle tool, or we can use Stretcher, or we can use various other different tools which are based on the Needle-Wunsch algorithm. Similarly, for local alignment, we have different tools like it is based on the Smith-Waterman algorithm. So there are various tools like Water tool, Matcher tool, L-Align, and S-Search 2S EQ tool for the local alignment of the sequences. BLAST, as the full form suggests, which is the basic local alignment search tool. So it is the tool which is used for the local alignment, and it finds the regions of local similarity between the sequences. This program basically compared a nucleotide or a protein sequence to a sequence database which is already present and calculate the statistical significance of the matches, whether we have obtained the similarity by chance or it is actually the similarity which is present in the genome. So it calculated that based on the statistical significance. And BLAST can be used to infer the functional and evolutionary relationship between sequences, as well as help identify the members of the gene families based on the based on the percentage similarity between the two sequences. So there are various forms of blast which are present. The one is a nucleotide blast, in which the subject is also a nucleotide and the query is also a nucleotide. Just a second, somebody wants to join. Okay. So the next is blast X, in which the translated nucleotide is aligned against the protein. The next is T blast N, in which the protein is aligned against the translated nucleotide. And the last is the protein blast, which is in which the subject is also protein and the query is also protein. So the question is, comparative analysis of protein sequences using the tools such as BLAST can help us to identify the rate of evolution of genes, identify protein families, or it can identify the phylogenetic position of the species being analyzed, and all of these. Okay, Vignesh has answered all of these. Anyone else who would like to answer? Okay, so let's move further. So the correct answer is all of these, as I explained clearly in this slide, that it can be used to infer the functional and evolutionary relationship, as well as it can help in identifying the members of the gene families. The next question is, which among the following options represents a group of data banks that can be used to analyze the nucleotide sequences? So is it Gene Bank, DDBJ, EMBL Bank, or it is PDB, or is it RCSB, or it is UniProt? So what is the correct option? Okay, option A. Anyone who would like to tell what is PDB, what is UniProt, if they are not nucleotide sequence, if they are not databases which are used, data banks for the nucleotide sequences? Okay. So the correct answer is option number A, that is Gene Bank, DDBJ, and a European Molecular Biology Laboratory. So these are the Japanese and European banks for the analyzing the nucleotide sequences. And UniProt or PDB, that is the Protein Data Bank, are the databases or the data banks which consist of the information about the structures of the protein and the other related functional information about the proteins. So, the next question is, often people use basic local alignment search tool BLAST to find the regions of similarity between biological sequences. The purpose of the BLAST tool is to compare queried nucleotide or protein sequence to the existing sequence database, and it provides the calculated statistical significance of the matches. Identify the best option which describes the different type of BLAST search. So nucleotide is for nucleotide to protein, blast X for nucleotide translated nucleotide to nucleotide, T blast N is protein to translated nucleotide, and protein blast is for protein to protein. Similarly, there are other options. So anyone who would like to tell, it's a very easy question. Right. So Vignesh said PDB protein data bank. Okay. So B. Okay. Anyone else who would like to answer? At least you can write it in the chat box. It will help you only. Okay, fine. So the next correct answer is, as Vignesh said, so nucleotide blast basically is for the nucleotide to nucleotide. Blast X is for the translated nucleotide against protein. T blast N translated protein against the translated nucleotide, and protein to Protein blast or the blast P is for the protein to protein. So the next question is shown below is a screenshot of the sequence alignment. The shown alignment represents what carried out by what program, using which program which we have carried out this alignment. So you can check what is query, what is a subject, and based on that you can tell easily what is the program which we have used and what we have carried out in between the subject and the query. Is the question clear to all? Okay, Vignesh has said option A. Would you like to explain it also? If you can unmute and explain it to all. Okay, that's fine, not a problem. So we can see in this figure the alignment which we have shown here. The query here represents the single amino acid residue representation, that is, and the subject is also the amino acid residue representation. So in this we can say by looking at the subject and the query that we are aligning a protein against a protein. So it is a protein sequence alignment, and the program for which we are like the program by using which we align the protein sequence are the is the blast P. So the correct answer is option number A, that it is a protein sequence alignment and which is carried out by a blast P program. So the next question is based on multiple sequence alignment. Multiple sequence alignment can help us to predict the descendants and the ancestors. It can identify the potential functional domain of the protein which are not yet characterized, or we can validate a mutation identified in a genetic disorder using the multiple sequence alignment, and all of these. Okay, option D. So, right, we can predict the descendants and the ancestors. We can also identify the functional domain of the proteins which are not yet characterized, or we can validate a mutation which are identified in a genetic disorder. So basically, in order to find the descendants and the ancestors, we can check the it is basically based on the similarities and the differences which are present in a sequence. So by identifying the sequence similarity, by identifying the conservation in a particular region of a protein, or what we say is a functional domain, we can say that since this region is conserved, therefore it must be or it might be functionally important. So we can check, there might be some regions which are highly conserved but whose functions are still unknown. So by identifying that those conserved regions, we can identify the potential functional domain of the proteins which are not yet characterized. Or also we can validate a mutation which are identified in a genetic disorder by checking, by aligning our sequence with the by aligning our sequence, as I said, of a healthy individual with a diseased individual. We can check whether there is a difference in any nucleotide or in any amino acid residues that is leading to that particular genetic disorder. So the next concept is the molecular phylogenies. So basically, it is based on the concept of the construction of the phylogenetic trees. The evolutionary relationship among organisms are summarized in the diagrams which are called as the phylogenetic trees, or we can simply call them as phylogenies. These trees may only show the relationship among the organism, or they may also show the timeline to indicate how these organisms have evolved from the ancestors. Now, knowing the phylogenetic relationship among the species is fundamental for any studies, for various studies in biology, because by knowing the phylogenetic relationship, we can have an idea how a protein have evolved, how we have evolved in the time being, and what are the things which are still conserved, what selection pressure we have overcome. So what are the functional importance of a particular protein, of a particular region in a DNA? So basically all these things are this molecular phylogenetic tree not only reveals the state of evolution but also reveals various different events which have occurred during the time of evolution. So an accurate phylogenetic tree underpins our understanding of the major transition in the evolution, such as the emergence of new body plans or metabolism, and where the new gene originated, detecting molecular adaptations, understanding morphological character evolution, and reconstruction demographic changes in recently diverse species. Also, by the comparison of genomes at different phylogenetic distances are appropriate to address the different questions which I explained in the above point. So, phylogenetic tree can be compared with a normal biological tree, which has a root, which has a branch, which has nodes, and which have leaves. The leaves of a tree here are also called as the tips, and these tips represents various species, populations, individuals, or even the genes, depending on the for what you are creating a phylogenetic tree. So the if the tips represent a formally named group, they are called as taxa. For example, vertebrates, invertebrates, or you can have an like mammals. So these are under the group which are formally named groups, which are known as taxa. So and a branch represents the persistence of a lineage through time and may subtend one or many leaves. Branches connect to other branch at the nodes, as we know. So the node is the point where one branch connect to the another branch, and which represents that it is the last common ancestors of the organisms that are originated at the tips, and this is the descendants of these. So suppose, for example, if my cursor is visible, so this is one node, and we can see that the organisms which are present at this node are the ancestors for all C, D, and E. So basically a branch connecting a tip to a node is called an external branch, as we can see here, and a branch which connects the two nodes is called an internal branch. Now what is a clade? Clade, to understand the term clade is very important in order to analyze any phylogenetic tree. So clades are the groupings on a tree that includes a node, that is this, and all the lineages which are present over here. So the set of all the tips in a clade is defined as being monophyletic, since monophyletic term actually refers to the fact that it includes all the descendants of an ancestral lineage. So basically both A and B, since they have a common ancestor which might be present at this node, so both A and B are clades of this ancestor, we can say. So in order to understand the phylogenetic tree, as I'll describe later in my slides, it is very important to understand what is a clade. So anyone who has any confusion regarding the basic concept of phylogenetic tree? If it is clear, we can move further. Okay, fine. So, think just a second. Okay, clear. Basically, phylogenetic tree can be unrooted or it can be a rooted tree. Unrooted tree, the first figure here, figure A, shows a phylogenetic tree which is unrooted. It only shows the relationship that whether A is related to B or C or D, if it is related or not. But the one which is rooted actually shows that from where the deviation has occurred. Like what is a common ancestor of B, C, and D? So this must be a common ancestor. The organism that might be present at this node are the common ancestors. And then how from B, C, and D evolves. So basically each bifurcation in a tree represents a common ancestor of the organism further out in the rooted tree. This is a generalized phylogenetic tree, and in this we show that with time this organism evolved and leading to the development in four species, that is A, B, C, and D. And the phylogenetic tree basically imposes to answer the following questions like what sequences account for unique features in an organism if they are so closely related. Similarly, what sequences show a signature of purifying selection that is are likely functional leading to the divergence at this level, and what is the core set of proteins in the multicellular organisms, since all of these must be having some common set of the proteins that might be present in all of these, and uh, that is similar to what their ancestors have. So now there are certain properties of a phylogenetic tree, and I'll describe them like they show the relatedness, they show whether how much they are related or how after, or they show a timeline also, which we'll discuss later. They show a timeline like at what point of time these species might have evolved from the previous species, and how much amount of evolution these species have faced. So trees can be depicted and modified in a number of different ways, as we can see in this diagram, regardless of whether a tree is oriented left to right, top to bottom, outward from the middle in a circle, as we can see in this middle figure, or along a diagonal, the key information remains the same. So, irrespective of what is the orientation of the tree, the information in the phylogenetic tree remains the same. So can anyone like explain how the information, although it is written on the slide, but if anyone would like to explain how the information in all these three trees, which are represented here differently, represents the same information? Like how will you analyze whether these trees have the are representing the same information or not? Okay, like there is a question how to understand closely or distantly relatedness using a phylogenetic tree. So, Ricky will come to this point later, and if still it is not clear, you can ask you can ask me in between also before I move to the another topic. So, I'll come to that also how to understand closely or distantly relatedness using a phylogenetic tree. So, basically, we can see what I introduced you to a concept of clades. So in this all these three figures, we can see we can find the different clades. So we see here like clades are what? The organisms which originate from a common node. So we can start from this, like D and E form a clade in this figure. In this figure, we can see that from this node, D and E are originating and thus are a part of a common clade. Also in this figure, we can see that from here, D and E are getting separated from each other and thus they are a part of a common clade. So in this way, you can check all these clades, and if they are giving the similar clade information, they are forming the same clades, we can say that the trees are identical to each other. So in these three figures, we can say that because in all the cases, the list of the clades is identical, thus the orientation and the style of the tree does not change the relationship it represents. So basically, the relationship which is represented by a phylogenetic tree does not depend upon the orientation or the style of a tree. You can use any kind of orientation or any style, but the important point is that the list of the clades should remain the identical in all the cases. Like it should not change in any of the cases. The next thing is also along the same lines, the nodes of the tree can be rotated or cannot be rotated without or whether it cannot be rotated. So the answer is that it can be rotated without modifying the topology. So we can see in this also we will see whether the clades which are formed are identical or not. So we see that we have rotated this right hand side arm of this phylogenetic tree, and but we can also observe that the clades which are formed in this, that is A and B is one clade, C, D, E is one clade, D and E are one clades. So same clades or the identical clades list of clade is also obtained in the right hand side figure in which we are getting three clades A and AB, ED and CDE. So the since the list of the clades is identical, information is remaining the same. Therefore, we can rotate the nodes of a tree without modifying the topology. But we should not modify the topology, because if we'll modify the topology and we'll rotate the nodes of the tree, then the information might change, as it may leads to some other clade formation. So there is a question which is asked in the chat box, that how will you identify the closeness or the relatedness of a species? So basically, there is a concept, like it is just a hypothesis that considering evolutionary changes to be constant, like suppose if we consider that a nucleotide is changing at a rate of like one nucleotide changes in approximately 1 million year, or let's say in 10,000 years. So we can say that by counting the number of changes, excuse me, there is someone who wants to join. Okay, fine. So by actually this is known as the molecular clock of evolution, and by knowing the amount of the, like by knowing the timelines, by knowing the similarities or the differences between the genetic sequences, and then creating a phylogenetic tree, we can have the branches which are different in their length, and by seeing the differences in the length of the branches, we can see whether these organisms are originated from the same, like at what time point these organisms have been originated. Okay, so Ricky, is that clear to you? You can answer in the chat box also if it is not. Hello, am I audible?
A confusion is there, but a little bit have if you can explain it one more time.
Okay, so what is your like where you are getting confused? Can you please explain it a little?
I understood the clades and all how they are related to a common ancestor and all, and it should be uniformed and it is stable throughout all the
Yes.
clades. So clades are could not differ from one to another, but the closeness and the distant relativeness uh, could be different. So the distance, how it can I think how it can change the relatedness amongst the species. That is where I am getting confused.
Okay, so I think when I'll discuss about the construction of a phylogenetic tree in my later slides, maybe it will answer your question in detail. But for now, I can say that while constructing a phylogenetic tree, I have not shown that phylogenetic tree here, but there are some phylogenetic trees which are constructed using the hypothesis where it is considered that the evolution occur at a constant rate, and uh, this is known as the evolutionary clock hypothesis, where we considered that amino acid or a protein, or we can say a nucleotide change, there is a change in a nucleotide or a amino acid happening at a particular, after a particular time. So after knowing the differences, by aligning the sequences using various tools, as I mentioned earlier, like BLAST or multiple sequence alignment, you can find the differences, the number of differences which are present in one sequence from the other sequence. And based on the number of differences which are present, you can say whether, suppose there is only one difference which is present in species A, and there are two differences which are present in species B. So considering that the evolution happens at a constant rate, we can say that the species A is more closely related to the ancestor, while the species B happen to be evolved later in this later during the process of the evolution. But the point is that although in this hypothesis we consider that the evolution occurs at a fairly constant rate, and therefore we can draw a figure where the length of the branch on a chronogram tells us the relative probability that a particular trait evolved on this branch at that particular time. However, we can also say that this will not be true when the rate of evolution is not clock-like. So there are like two thoughts, like whether it is in a clockwise manner or it is not, or whether it is happening in not in a clockwise manner, like it is not clockwise. So when you are like when you want to see whether the how the branch length is demonstrating the species relatedness or distance, you can see that this you can for constructing such phylogenetic trees, you can use that molecular theory in which like evolutionary theory in which states that the evolution is a clockwise event. Also, like from normal phylogenetic tree also, you can identify whether a species is related or distantly related by checking the nodes. For example, the organisms which have, like in this figure, as I said, so this phylogenetic tree answers various questions. For example, suppose here is species 0 which is present, and during the course of evolution it evolved into various species like A, B, C and D. But there are some genes that might be present in species that must be present actually in species 0 as well as in this A, B, C and D, which are conserved during the time of evolution. So by actually noticing the differences or the similarities in the protein sequences or the nucleotide sequences, you can say that if two sequences are highly similar to each other, we can say that these two organisms are closely related as compared to the one in which the protein is present but the sequence similarity decreases. So, is that clear?
Can you please present the previous, the one which you have
Okay,
shown,
okay,
the
okay,
first the figure, and using
okay.
that, can you please one more time repeat the
Do,
closeness and the
like,
distant relatedness using this? Yes, this simple diagram.
okay. So, like basically what we can see is that suppose here is species 0, right? So
Yes.
this species then evolved into some other organisms, right? There must be some organism which must be present at this node, and then it evolves into another organism which is D, fine? So this species which is present at this node further evolved into other species like A, B, and C. So by comparing the sequences of, suppose I compare a sequence of any X protein, and I found that the percentage similarity, what I will find is the percentage similarity of this protein. What I expect is that the percentage similarity of in the sequence of this protein will be more in species A and B as compared to the species A and D, as during the course of evolution, there might be some changes which have been accumulated in the genome of these organisms. So, since this organism evolved, we cannot say that it evolved for example, since this organism evolved from the organisms or the species which is present at this node, so it must have accumulated more changes. So, we can say that A and B must be closely related as compared to A and D, right? They must definitely be having some set of genes, some set of proteins which are common because they also have arrived from the common ancestor, which is species 0, as I was mentioning. But there must be some differences, but the differences have accumulated during the course of evolution, and thereby the species A and B must be more closely related if we compare the sequences of both A and B. But on the other hand, if I compare the sequences of A and D, I'll find the percentage similarity decreases. Is it fine?
Yeah, it's completely clear now.
Okay, thank you. So there are two terms which are cladogram and phylogram. So the left hand side figure represents a cladogram and the right hand side figure represents a phylogram. These figures are the representation in this figure is based on the by considering the figure are generated by considering that the rate of the evolution is not clock like, that is it is not fixed, and the evolution may occur at any point of time. So the branch length here basically represents not the evolutionary distance, but the evolutionary traits which they have accumulated during the course of the evolution. For example, and there is a difference between a cladogram and phylogram, which I'll discuss in this slide. So trees may just show tree topology in which they are called as cladograms. Sometimes in trees called chronograms, branches are scaled to represent the opportunity for evolutionary changes by drawing the branch length proportional to the time. As I was mentioning, as what I was trying to explain it to Ricky, that in some cases we draw the branch length proportional to the time. And if we assume that, and this only happens when we assume that the evolutionary changes occur at a fairly constant rate, the length of the branch on a chronogram tells us the relative probability that a particular trait evolved on this branch and about how many years ago. However, this will not be true when the rate of evolution is not clock-like. So in that case, we can show instead of phylogram, a tree whose branch length are drawn proportional to the expected amount of trait evolution. So basically, we can see that in a cladogram, you can see that all the tips end at the same pace, but in a phylogram, on the other hand, the tips ended at a different pace. So because in a phylogram, we consider that the branch length shows how much trait, like how much trait like amount of trait it has accumulated during the course of evolution. Like for example, what I mean to say by this is, here we can see that the branch length for the F is maximum as compared to the branch length of E or branch length of D. But we cannot say that the F evolved much better as compared to D or E. It just shows that the F evolved, like from this node, F evolved earlier, but and it acquires certain traits, and amount of traits which it has acquired during the evolution is shown by the length of the length in this phylogenetic tree. So similarly, the from the time of originating of this D and E, how much traits, how much different traits it has acquired during the course of evolution is shown by the branch length D and E. So in a chronogram where all tips are living, all tips are equidistant from the root, since the branches represent the absolute time since divergence from a common ancestor, while in a phylogram, in contrast, different tips may be present at different distances from the root, indicating the expectation that some tips may have accumulated more traits than the others during the time of evolution. Any doubts? It is very important to understand what is a cladogram, what is a phylogram. So, if there is any doubt in between this, okay,
Um, Here I can say that if you can look on the two diagrams which you have
yes.
which you have presented now, so which are the which how we can detect the common ancestors here in this in these two figures?
So basically, the common ancestors are represented at the nodes. So there must be some species from which these all, like from which this A has been, like for example, in order to make it easy, we can consider this node. Wait, is my slide visible? Okay. We can consider this node, and from this node, from this species, various other species evolved during the evolution. And so this is the common ancestor for F as well as for A, B, C, D, E. But if I talk about D and E, the common ancestor will also include the species which are present at this node. But if I talk about C, D, and E, the species which are present at this node will not be the common ancestor for C. But definitely the species which are present at this node will be the common ancestors for C, D, and E. So basically, the common ancestor, if you want to look at, you can check the node from which these different species or from which these different tips are originating. So this node depicts the common ancestry of all the species, all the tips, all the taxa which have originated from the common ancestor. Is it clear?
Yeah, it's better now.
Okay. Still, if you have any doubt, you can ask.
So, is there any name? Why can't we point out like A, B, C, D, the common ancestors here? Or if we can do it, so how we can do it?
Like, sorry, I didn't get your question. How can we
I think
say?
and these commons are actually you said that for detecting the common ancestor, we need to find out the nodes. So as we have seen the taxa, you said A, B, C, D, E, F, right? So we have pointed it out like that. So how is there any problem, or can't we point out the ancestors by naming or by symbol or anything else like that?
Definitely you can. It is just a representation. A, B, C, D, E is just a representation. You can actually give the name of the species, name of the taxa, which we are which you are discussing. And I think this will be more clear in my next slides. One of my slides had that such kind of phylogenetic diagram. I'll ask you at that slide if it is clear or not. And still, if it is not clear, I will try to make it more clear to you. Right?
Thank you.
So. So basically, this is the slide. So there is a concept of orthologs and paralogs. Orthologs and paralogs are the genes which have the common ancestor, but during the course of evolution, either by the process of the speciation, that is the formation of a new species, or by the gene duplication, these different genes have formed which have a different functional role. So basically, if I'll see orthologs are what? Orthologs, for example, here we have considered the ancestral nematodes. So fine, this is one of the nodes. That is there must be some nematode which is the ancestor of both the specifers and elegans. So basically, this ancestral nematode has a gene which is DAF16, and by the speciation event, this DAF16 passes to both the species, which is the Pacificus and the C. elegans. While in case of paralogs, what happens is there is a gene, for example, DSH1 gene, which is present in ancestor. Speciation event happened. Speciation event leads to this DSH1 gene in both Pacificus and C. elegans. But after that speciation event, there is another event which happened in C. elegans only, which is a gene duplication event that leads to the formation of two kinds of genes, which is C. elegans DSH1 and C. elegans DSH2. So these two genes, which are formed or which have evolved during the course of evolution as a part of gene duplication event, are known as the paralogs, while the genes which have occurred as a result of the speciation event are known as the pacificers. Are known as the orthologs, while the genes which are occurred as a result of the speciation event in both pacificus and C. elegans are the orthologs. So, Ricky, is that clear? Like we can also name, like you can see that figure C also, where based on the percentage similarities between these genes, we have made a phylogenetic tree. Considering that they have originated from a common ancestor, these like both Pacificus and C. elegans have originated from a common ancestor. How did we come to this conclusion? We come to this conclusion by finding the similarities in between a gene DF16, which is also present in Pacificus and which is also present in C. elegans. But then how this C. elegans so this is a phylogenetic tree representing the different genes. So now this C. elegans DSH1 is more closely related to C. elegans DSH2, which has originated in the same organism as a part of a gene duplication event. So this is the basically the multiple sequence alignment diagram in the figure D, where I have shown a 34 amino acid residue window of the protein alignment, from using which the figure in the part, that is the tree in the part C, has been generated. And basically it shows the sequence conservation of DSH1 orthologs and the subsequent divergence of DSH like C. elegans DSH2 from the other sequences. A similar example is the hemoglobin and the myoglobin. So hemoglobin in humans is orthodox to hemoglobins in chimpanzees, but the myoglobin in human is parallel to the hemoglobin in humans because it is supposed to originate as the part of a gene duplication event during the course of evolution. So, is it clear or any doubts?
So in this diagram, we can see there that it is quite like it is well defined, and you have told that the orthologs and paralogs could be defined easily from looking at the diagram. So if you can look out, if you can see the only the tree, so there would be two paralogs and one ortholog, or two are ortholog. How many orthologs would be there in
How
this
are you
diagram?
saying like okay, so you tell me which are the orthologs and which are the paralogs?
Paralogs are the C. elegans, DSH1, DSH2. These are the paralogs, and
Paralog, right.
yeah, and orthologs are the one called DSH1 Pacificus uh,
The C.
and
elegans DSH1 basically,
yeah,
right.
yeah, yeah.
So you
So
want to like how will you say? So the point is there must be a common like the see you can see this node where this gene duplication event is happening, right? So
yes,
there is some gene which is present in at this stage. So the gene which is present at this stage is is undergoing gene duplication and leading to a formation of two genes in which the one is similar, which is DSH1, and there are some modifications in the nucleotide sequence leading to a formation of a different protein, and thereby leading to a formation of a different set of gene, which is a C. elegans DSH2, right? So this gene, which is present at this node, which is actually the ortholog for the P. pacificus DSH1. Is that clear?
yes,
Okay. I hope it answers your question now. Like, जो
yes,
भी doubt ना, okay, fine.
yes, huh.
So basically, constructing a phylogenetic tree involves these steps, which I have already mentioned. Like, firstly, we will assemble the data from various species. We will we will first identify the contaminants. The contaminants may be, for example, in our system, if we will see, so there are gut microbes which are present. There are many other commensals where there are various symbionts, there are various parasites which are present, or there might be some experimental errors that have happened during the data assembling. So we will first we will first identify the contaminants and we will remove the contaminants. Next, what we will do is we will do the all against all comparison because we don't know what is similar to what. We will do all against all comparison using BLAST, or we can use some other tools, and based on that we can identify the sequences that are homologous based on the percentage similarity between all the species of interest. For example, if I want to see in between the whether humans are related to fish and Drosophila, so I can go for the uh, sequences of these three organisms only, or if I want to add more, I can take the more sequences. Now, after getting the alignment, the sequences of the putative orthologs are aligned to generate a multiple sequence alignment. After this, the MSA, we can remove the outliers by checking for the paralogs. So the MSA can be analyzed to produce an initial phylogenetic tree, as I mentioned, for the putative orthologs, which can be used to identify the remaining paralogs by seeing the percentage similarity between the other proteins. So therefore, by using this ortholog and paralog concept and by using the local alignment and multiple sequence alignment, we can finally come to a alignment by we can finally the MSA is then finally typically filtered to remove the regions of unreliable alignment, and then finally the orthologs are concatenated and which result in the production of a phylogenetic tree, which we see that how the different taxons are related to each other. So one of the important concepts which leads to the which is important during the course of evolution as we see is the speciation. Speciation is the process which leads to the formation of a new species. Species according to the biological concept of species, different species consist of the organisms that cannot have a sexual reproduction among the organisms of the other species. That is, if there is a species A and there is also a species B, we can call them only two distinct species only when the individuals from species A cannot reproduce sexually with the individuals from the species B. So, what are the different types of speciation events that have happened during the course of evolution, or what are the different types of speciation events that can happen? Okay, I'm so sorry. Uh, so, basically, what are the different kinds of speciation that may happen? The first is the allopatric speciation, and it is a very common form of very common process that leads to the speciation. So, suppose there are individuals, there are a population of an organism that inhabits a territory. Due to some geographical barrier, the population is divided into two subpopulations. Let's say a river started passing through that from a forest, and then the individuals from one side cannot move to the other side. Thus, during the course of evolution, the separated subpopulation changes accumulate some genetic changes. And when these genetic changes, which are accumulated in a population, result in the reproductive isolation, then it leads to the formation of a different species. And later on in the life, when they are reunited, the subpopulation now cannot change the genes, and therefore they have entirely formed or evolved into a different species. By saying that they have evolved from this common species, we should always keep in mind that it is not like that the previous species or the ancestors do not exist. It might be possible that they do not exist, but it might be possible that the previous species or the ancestral species also exist, but it has led to the formation of some other various species during the course of evolution. So this is a kind of allopatric speciation where a geographical barrier is introduced and thus leading to the division of subpopulations which acquire genetic changes. The second is the sympatric speciation. So in this, a population of organism inhabits a territory, and without any geographical barrier, some subpopulations form in the absence of any geographical barrier, and they begin to differentiate genetically. Like they start accumulating some genetic differences. Ultimately, these genetic differences accumulate so much that they cannot that this leads to the reproductive isolation within the same area only, and each subpopulation now become a distinct species. So, apart from this, the speciation can also happen in other two ways, which is the parapatric and peripatric speciation. So, in peripatric speciation, the new species are formed in isolated smaller minor population, as we can see here. So the new species are formed in an isolated minor population, and this minor population are refrained or are prevented from exchanging the genes with the main population. Now, suppose from this pet like this from original population, a bunch of population get separated, and this population now do not reproduce or evolve to have the reproductive isolation, and now it will form a different species among themselves. Similarly, in parapatric evolution, in parapatric speciation, there is no specific extrinsic barrier as we see in case of allopatric speciation, but the population is also continuous. But the population does not mate randomly. Individuals are more likely to mate with some particular individuals which are present in the vicinity of their geographic region, and due to which the two different individuals which are although not geographically isolated but still they are present in distance with each other accumulate different genetical changes leading to the reproductive isolation, and thereby new distinct species are formed after equilibration of the new ranges. So the next question is, which one of the following options is incorrect with regard to comparison of sequences of two distantly related species? Protein sequences showing very high similarity between the two species are more likely to have similar function. Protein sequences that show least similarity between two species are less likely to be functional. Protein sequences that is present in one species and absent in the other indicate its loss in the later if it is a phylogenetically recent species. And the next option is all of these. So which is incorrect with regard to the comparison of sequences if we are comparing the sequences of two distantly related species? What is the answer? Does anyone like to answer this question?
I can say that A and B are correct statements, but C, I think once.
Okay. So a protein sequence, we know that very high similarity are more likely to have a similar function. Similarly, protein sequences that have a least similarity between two species are less likely to be functional, right? The next option is a protein sequence that is present in one species and absent in the other indicates its loss in the later if it is a phylogenetically recent species. So, basically, if we are comparing the sequences of two distant related species, the correct answer, the incorrect point is option number C. The protein sequences that, uh, I'm so sorry. The correct answer is option number C, option number B, because the protein sequences that show least similarity between two species, it is not necessary that they are less likely to be functional, but we can say that they are less likely to be functional in the other species. We cannot say that they are not functional, right? If they are present, it means that they are functional. They might have acquired a different role, they might have acquired a different function, but if they are present in both the species and since the presence itself indicates that the proteins are functional, right? Is it clear?
I chose the incorrect option.
Yeah,
Sorry.
it's okay. The next is given here is a generalized phylogenetic tree derived by the comparison between genomes of four different species named as A, B, C, and D. So now if I want to identify the genes with conserved functions in species A, if I want to see what are the functions of a gene, conserved functions in a species A, I for what species I should analyze the sequences, for what species I should analyze the genome sequence of. So should I analyze the genome sequence of a species A only, or should I acquire the genome sequences of species A and B, or A, B, C, or all four A, B, C, and D? So the correct answer is that if I want to see what is the gene which is conserved in species A, I should analyze the sequence of all the species A, B, C, and D, and then I should have alignment of those genome sequences. And by checking the percentage similarity, like which is the region which is highly conserved, I can say that if it is present in all these species, like in A, B, C, or D, this means that the region of the gene is highly conserved because we can see that the species have evolved later in the evolution. So we can see that if it is present in all these species, therefore it must be highly conserved. The gene must be highly conserved and its function must be very important. The next question is, a group of students were asked to analyze the genome sequence of four distinct species, which are QPRS, and they are asked to plot a phylogenetic tree to predict the evolutionary distance between the species. Now, four students used different approaches and came up with four different phylogenetic trees, which are named as H, I, J, and K. The instructor felt that the prediction given by one of the students did not meet the expected result. So, which one shows a different evolutionary history from the others? Does anyone who would like to answer this?
K.
Uh,
Okay, I don't know.
okay, K. Why do you think it should be K?
Uh, I think it is of equal distance, so it should be different because it is or arranged in a organized manner, and it is descended like in the each and every species have evolved uh, sequentially, not evolved randomly. I think maybe I'm wrong. I don't
Okay.
know.
So Vignesh has answered option number like three, which is J. So one of the concept I introduced was of the clade, and if two or three trees, doesn't matter how we have drawn that, if all the trees are having the same or identical list of clades, then these trees are said to be identical. So can like Ricky, can you now check what should be the answer? Like why it should be J now? Like Vignesh has answered it correctly. Did you understand the concept of clades? Clades are the organisms which have originated from the same species, from the same organism, right? Okay. So Tagore has also answered option J, correct. So in all the three, like in H, I, and K, we can see that the list of the clades is identical. For example, in H, it is QPRS, it is PRS, and it is R and S. Similarly, in this I, it is QPRS, which have one common ancestor. Then second is PRS, which have another common ancestor, and then RS and R and S, which have a different common ancestor. So, but in case of J, we'll see then PQRS are having a common ancestor, but here the P and S have a one common ancestor, but in the last point we can see it says that P and R have a very closely related one common ancestor, but what we see in the other figures is that R and S have a one more closely related common ancestor. So is that clear? It doesn't matter whether how you are drawing the phylogenetic tree, but the information which is provided in this H, I, and K is similar. But the information which is provided in J says that P and R have originated from one common ancestor after originating from after when this common ancestor have originated from some other common ancestors. Vicky, is it clear now? Why it should be J?
Oh my God.
Is it clear? Okay, fine. So, one thing I want to ask, do you want me to continue with the questions or like do you want me to end the session? If anyone who would like to answer in the like can tell me in the chat box if they want me to continue with some more questions. If it is okay for all of you, I can continue. I don't have any. Okay, fine. If English want me to continue. The next is in the phylogenetic tree, which is given here. All the four species being analyzed are the mammals. Now, amongst these species, A and B can fly, C is the terrestrial, and D is a marine species. Now, my project is to identify the recent genes that helped in the evolution of mammals to fly. For this, like I would analyze the sequences of both species A and B only, species A, B, and C only, species A, B, and D only, and species A and B only. So, anyone who would like to answer this question? So, it must be species A, B, and C only. Since D do not have any role in this flying, so do not have any gene for the flying. Similarly, although C do not have any gene in the flying, but the but it has a common ancestor from with both A and B, after which only, like, there must be some changes during the evolution which have happened in that organism which is present at this node, that leads to some changes in the organisms of A and B that help in acquiring them the capability to fly. So if I'll compare the genome sequences of A, B, and C, I'll get the idea like what is the gene which is differing in both AB versus C, and thereby giving the ability to fly in A and B while the C still remains the terrestrial. So the next question is, which one of the following distinguishes the field of comparative genomics from genomics? Option A is use genomic knowledge to understand an organism. It compare various genomic approaches and develop novel methods. Use information from many genomes to learn more about the individual genes. And option D is all of these. Anyone who would like to answer? Okay, option D. So basically, is you genomics uses the knowledge to understand an organism. But what the question asks is, what is the statement that distinguishes the field of comparative genomics from genomics? So, comparative genomics uses the information from multiple organisms, like as we see in case of multiple sequence alignment, or even if we are using local alignment, so by BLAST. So, we uses the sequences of multiple species, and then we use this information from many genomes to learn about the individual gene, whether this gene is conserved, this gene is not conserved, the function is conserved, whether this gene is important. So, option A is definitely correct for genomics. Option B is also correct, like we use various genomic approaches to develop novel methods using the knowledge of genomics. But the distinguishing feature of comparative genomics from genomics is that the comparative genomics is the field of genomics which uses the information from many genomes to learn more about the individual genes. The next question is, we have sequenced the genome of three species of butterfly named as X, Y, and Z, and analyzed the genome for the presence or absence of the four genes which are R, S, T, and U. So these are the three different species which are analyzed for four different genes. Based on their presence or absence, I would like you to draw, like, which is the I would like you to predict which is the correct phylogenetic tree depicting the information which is present in the table. So for example, gene RSTU is present in species X, but in species Y gene R is absent, and in species Z gene R, S, and T is absent, only gene U is present. So what is the correct option, which is the correct phylogenetic tree? So the correct option is option number A, because we can see that species X and Y are more closely related, while species Z is not that closely related, by checking the presence or absence of the different genes. Like in species Z, only gene U is present, while in species X and Y, most of the genes are common. So, because they share more genes, species X and Y are more closely related, and hence they are likely to be closely related, then as compared to the species Z. So, the correct answer is option number A. Now, this is the complete summary of what this question summarizes of what we have studied today. So basically, we have decided to follow the lead of Craig Venter and James Watson, and I sequenced the genome. The first 100 nucleotides of my sequence has the following sequence. And what is the function of this? How would I find the function of this DNA? How would I get the information on what chromosome is it located? Is the sequence unique, or are there similar sequences which are present elsewhere in your genome? The next question is this sequence present in the genome of other species also, or is it unique to yours? genome only. So how can we go with this? Anyone who would like to elaborate this and who would like to give the answer for this? It is a like this is kind of summary of what we have studied till now. So what we will do is firstly we will do a blast of human genome transcript sequences in the gene bank nucleotide database that inform us that the 100 nucleotide sequence is a part of human. For example, it informs us that the 100 nucleotide sequence is a part of the human globin gene, and this human globin gene is present on the chromosome 11. This much information we get firstly by performing a blast against you blast search of the human genome transcript. Fine. Firstly, we find on what chromosome it is present. Next is also we find the function that it is a human globin gene, and then we can go for the then we can further study the function of this gene. The next is the 100 nucleotide sequence is identical to the sequence of one strand of the human globin beta gene. And the sequence is also very similar to the sequence of another gene, which is human alpha globin gene, which is located adjacent to the beta globin gene. So, therefore, we get the answer that the sequence is not unique. There are similar sequences which are also present in our genome. Now we will perform the blast search of all the NCBI genomes that shows the sequence difference of only one nucleotide, as I mentioned, and it shows that the same sequence which is present on the chromosome 11 in humans is also present on the chromosome 11 of chimpanzees, and only there is a 7 nucleotide difference, and it is present on the chromosome 14 of rhesus monkeys. So this gives the information that the sequence is also present in the genomes of other species. So from this we can infer that clearly the sequence of the globin genes are highly conserved in the all primates, since it is present in all the primate species. We can further go for the we can further expand this study, and then we can have a detailed analysis that whether this globin gene is present in all vertebrates or all in other vertebrates or not. So by checking the conservation, whether if this gene is conserved in vertebrates also, we can say that this gene is present in the vertebrates as well. So any doubts in this question, particularly the answer in this question, answer of this question? Uh, can please someone please tell me in the chat box if they have any doubt in this question? Is it okay? It is clear. Fine. So the next is it is well accepted now that mining of genome sequence data leads to new evolutionary theories that were difficult to comprehend earlier. So, for any such bioinformatic projects, there are three critical elements. The first is to three critical elements, that is, we need to find patterns, we need to make a specific observation, we need to come up with some hypothesis. So, what is the order in which these steps are followed? Do I need to first observe the sequence? Do I need then find the pattern in the sequence and then come up to the hypothesis? Or firstly, I should hypothesize something and then find the pattern and then observe the sequence? So, what is the correct chronology of the steps which we should follow? So the correct answer is first we should observe the sequences, then we should find the patterns. Patterns are the blocks or the segments of the nucleotide or the protein sequences which are similar, which are identical, which are similar in different species. Then we can come up with the hypothesis that since this protein is this sequence is highly conserved in all the primates or in all the vertebrates, we can hypothesize that this might be having a very important functional role in one of the properties which vertebrates show or which primates have. So we can then come up with the hypothesis, and then we can go for the experiments, and then we can check for our hypothesis basically. So this is the summary of overall functional genomics course and the different functional genomic techniques that we have discussed. We see that the DNA leads to the formation of RNA, protein, and then there are different metabolites which are involved in maintaining the health of an individual. Now, if we are checking it at a DNA level, it is genomics. We are checking it at an RNA level, it is transcriptomics. We can, if we are checking it at a protein level, it is proteomics. And if we are doing metabolite analysis, it is metabolomics. And these are the different techniques which we can use at a different level. For example, we discussed in detail about the PCR, we discussed about sequencing methods, we discussed about microarray. So these are the various techniques which we can use to in order to understand the function of our genome, and therefore they are very important for the functional genomics. They are important approaches for the functional genomes. So this is just a summary of various functional genomic techniques with certain advantages and disadvantages. Many of Some of these techniques we have not discussed in detail, but you can definitely go through these techniques, like for example, fluorescent in situ hybridization, which is a very important technique, and it is able to identify the it is able to identify the known abnormalities which are present in the chromosome. So it can help in identifying the chromosomal abnormalities. It can help in identify the translocations, whether certain part of a chromosome has translocated and has merged with another part of the chromosome. Similarly, RNA-seq. So RNA-seq is the sequencing of the complete RNA that helps in the that helps in having the information about the non-coding RNAs also. Similarly, we can have the microarrays. So these are the all these different various techniques with certain advantages, and all of these techniques have definitely have some disadvantages also. So by depending on our problem, depending on what we want to discuss, depending on what is our question which we are proposing, we can use these multiple techniques. And therefore, I would like to end this session with just one or two last questions. The table here shows the popular functional genomic approaches in this column A, and the possible objectives in the column B. So, which option identifies the which is correctly matching with what? So, I am telling the answer. The RNA-seq is helping in the alternative splicing, since RNA-seq have the data of all the RNAs which are sequenced. So by alternative splicing, if we are getting some different forms of mRNA or we are getting mRNA variants, we can have that information using the RNA-seq. The second is option B, that is FISH, is as I mentioned, it helps in the translocation studies, translocations in the chromosomes. The third is ChIP-seq. ChIP-seq, as I already told, it helps in finding the regulatory regions to which the promoter or the repressor are binding, and thus regulating the gene expression. And the last is the microarray, which helps in identifying the differential expression of the genes by having a fluorescent signal based on the intensity of that fluorescent signal, we can check whether the gene is getting expressed or not expressed. The last given below are some important target sets for the Human Genome Project and beyond. Some goals are met and some are yet to be achieved. So identify from the following options the sequence that correctly arranges their chronological implementation. So the correct answer is option number B. The correct answer is option number D, in which state that firstly we understand the structure of the genome, then we understand the biology of the genome, then we came to understand the biology of the disease, because now we have the knowledge of the genome and the genes which are responsible for causing that disease, or the proteins which are non-functional getting non-functional and leading to that disease. The next is the improving the effectiveness of the healthcare by advancing the sciences of the the next is to advance the science of medicine that leads to the improvement in the effectiveness of the healthcare. So last question is based on the transposable elements. Transposable elements are the elements which are the moving elements, and they get interspersed into the human genome. So which one of the following is not a transposable element of the human genome? So it is the IS elements. IS elements are present only in the prokaryotes in the bacteria, while all other transposons like DNA transposons or lines and signs are present in the human genome. So with this, I would like to end today's session, and I would like to thank you all for attending this. Still, if you have any doubt, you can post the doubts on the discussion forum. And we are now closing this functional genomics course. All the best for your exams. And thank you for your patience. And any doubts, thank you, Vignesh. I hope you like all the doubt sessions. You can post the feedback also if you want. The feedback link is provided. So, and still, if you have any doubts, we'll not have that doubt clearing sessions. But still, if you have any doubt, you can go and visit the YouTube link, and then you can post your doubts in the comment sections, or you can directly ask your doubts in the discussion forum of the functional genomic course page. All the best for your exams. If there are no other doubts, I would like to end this meeting. Okay. Thank you all for attending this.